Right, today's lesson, you're going to be looking at investigating the rate of photosynthesis. So you want to write your title down, which is investigating the rate of photosynthesis, your date today, and you might want to write down these keywords as well in your books. Okay, so if you want to pause the video and do that, and then we'll go through what we're doing. Alright, so when we're ready, we're going to look at today what different factors can affect how fast photosynthesis is happening, or what we call the rate of photosynthesis. And we're going to look at how we can measure that as well. So we're going to look at what we would do in a practical. And obviously, because we're not in labs this year, we're not going to be able to do a practical, but hopefully we can show you what one would look like. So, we'll start off with a couple of questions. So I want you to have a quick chat about this. So just think fair share. So if you think for yourselves and then have a minute to chat with each other and see what you think, and then I'll go through the answers. So what is meant by the rate of photosynthesis, and what conditions may affect the rate of photosynthesis? So if you think about what plants might need for photosynthesis, okay, and how that might affect the rate of it. Okay, so if you want to pause the video and have a quick chat about those two questions again. Right, and when you want to start again, we'll give you the answers for that. So the rate of photosynthesis is how quickly photosynthesis takes place. So it's rate of means basically how much is happening, it's how fast it's happening. So it's how quickly photosynthesis takes place. And the conditions that affect this are going to be light, temperature, and carbon dioxide. So light and carbon dioxide, obviously, because we know that plants need light and they need carbon dioxide for photosynthesis. Now, water is not usually a limiting factor in photosynthesis. Plants have usually got enough water for as much photosynthesis to happen as they need, so that's not really a concern. And temperature is because it is a chemical reaction, and if the plants are not at a warm enough temperature, they go a lot slower, just like a lot of animals do. If they don't keep the body temperatures up, then the processes happen slower. So plants are the same. So what I want you to do now, again, if you pause the video and answer these two questions in your books, just summarising what we've just talked about there. So what is meant by the rate of photosynthesis, and list the three factors that might affect photosynthesis. So pause it now. Okay, and we'll move on when we're ready. Right, so have a look at this picture here. Okay, we've got this equipment and I want to investigate how does light affect photosynthesis. Okay, so you've got this equipment, we've got a lamp, we've got some pondweed in a beaker of water, okay, and we can see what that's doing. We've got some transparent plastic to decrease heating from the lamp, so that's going to mean that temperature is not going to affect anything. And we would also have a meter ruler. Okay, so that'd be the only other equipment that we've got here that we don't have shown. So I want to discuss on your tables in just 2s or 3s, how would you use that equipment? Okay, so if you want to pause the video, have a discussion, and then I'll come back and we'll talk about what you would actually do. Okay, so when we are ready, if you think about this, little hint for this, oxygen we know is a byproduct of photosynthesis, so oxygen is produced. So how could we measure how fast the oxygen was produced? So if you look at the pondweed, there's a little clue in the picture here, specifically in this bit here, okay? So if you have a look at this bit here, we've got some little bubbles being produced. Those little bubbles show us what the the oxygen being produced during photosynthesis. So we can see that we've got bubbles being produced during photosynthesis, and that would tell us how fast photosynthesis is happening. If there's lots of bubbles, it's going to be lots of photosynthesis. Not many bubbles, not much photosynthesis. So if we have a look at what we would actually do in this investigation then, okay? What we would do is I would take my lamp and I would move it a certain distance away from my pondweed, okay? So I might start off, obviously this would be a meter ruler in real life, but on mine I might start off at 15 centimetres, then maybe do 10, then 5, okay, to get some different results. And what I'd be expecting to see is that the closer I moved it, okay, the higher the light intensity would be, okay. So if I move my lamp up close and just 5 centimeters away, then I'm going to get lots of bubbles being produced over here. So at 5 centimeters, I'd be expecting lots and lots of bubbles being produced from my plant, okay. If I then move my lamp though and moved it to say 15 centimeters away, I'd still expect to see some bubbles, okay, but just a few, okay. And I could count those bubbles. So I might count how many bubbles in, let's say, 1 minute. Okay, so I have got an independent and a dependent variable. So my independent variable, if you remember, what word we use for that, we use the word chin. Okay, because we change our independent variable. Okay, so we change the independent variable. And what we change in each time is the distance that the lamp is from our plant. So we're changing the light intensity. Now, when you're actually answering this, what you would want to say is we are changing the light intensity by moving the lamp closer. So what we're actually changing is not just the distance of the lamp, we're changing the light intensity. We're changing how much light we've got by moving the lamp closer or further away. Now, think about what we are our dependent variable, and if you remember on this one, it's mode. Okay, because we measure or observe our dependent variable. So what we're measuring or observing is our number of bubbles. Okay, so we're measuring our number of bubbles coming out, and that tells us the rate of photosynthesis. So again, what we're actually wanting to measure is the rate of photosynthesis, and we're doing that by counting the number of bubbles. So just like with the independent, we change the light intensity by moving the lamp further away. We are measuring the rate of photosynthesis by counting the bubbles. Okay. Now, last thing we've got is our control variables. Now, control we keep the same. So we want to keep the same those other factors that affected photosynthesis, so your temperature and your carbon dioxide levels. So carbon dioxide levels is easy, okay, it will just stay the same by itself, but you might want to mention that you want to keep that the same. We'd also want to keep the temperature the same, and this is where this sheet comes in. Okay, so from the lamp, we've got light coming through, which the light is going to be able to pass straight through the plastic sheet because it's clear and get straight through to our pondweed. We've also got lamps also give off quite a lot of heat, but the heat is going to hit the plastic and it's going to get absorbed into it and not get through. So it's not going to affect how fast the photosynthesis is happening when it's closer. Okay, so we are, again, this is what we're trying to get better answers. They say we're doing this by doing this. Okay, so we're keeping the temperature the same by putting the plastic sheet in front to stop the heat from the lamp getting through. You might also say that we're keeping the light in the room the same, and you might turn off all the other lights apart from this one so that you know the light in the room is not affecting the photosynthesis. Okay, so a couple of things that you could do there. So what I want you to do now, and if you need to go back and play the video again, feel free to do that and over the explanation of what you should be doing. I want you to answer these questions. You want to pause this for 10-15 minutes while you go through this. I don't know how long it's going to take you, obviously. But I want you to describe how to set up an experiment to investigate how light affects the rate of photosynthesis using the equipment shown here, okay, as well as a meter ruler. So I want you to draw the diagram and list the equipment you need. So you've got obviously your lamp, your plastic barrier, your pondweed in your beaker. You might need as well, if you're going to count, for 1 minute, how are you going to count for 1 minute? What equipment would you need for that? And how are you measuring the distance as well? So think about how you're going to measure these things that you're going to measure. I'm going to state your variables, so independent, dependent, control. And again, remember, we want to say what is it? So like light intensity is our independent and we measure that by moving the lamp away. We change that by moving the lamp further away. What is your dependent variable? How do you measure it? What is your control variable? How are you keeping it the same? Try and be specific. And then I want you to go into a full method, so it should be a full big paragraph describing how you, or even more than one paragraph, describing how you will carry out the investigation, okay? And if you're done, can you predict the results of the investigation? So if you want to pause now. Okay, when we're ready, we can move on. So I'll go through the answers really quickly. So obviously a diagram and your equipment, you've got everything you need on there, as well as we would also be needing, you're going to need everything that you see on the screen plus a ruler, okay, preferably 1 meter, and you're going to need a stopwatch because you're going to be timing the number of bubbles in 1 minute. Okay, right, so we talked about your variables already. Okay, so we've got our independent variable. Oh dear, so sorry about that. Our independent variable is going to be the distance of the lamp. Okay, our dependent variable is going to be the number of bubbles in 1 minute, or even better, our independent variable is the light intensity, which we change by moving the lamp further away. Our dependent variable is the rate of photosynthesis, which we change by we measure by counting the number of bubbles. And our control variable is going to be a couple of different things. I'm not going to write this all on, there's not going to be enough space. Our control variables are going to be the temperature, which we're going to keep the same by using the plastic, and we are going to try and keep the carbon dioxide levels the same, and also the light in the room the same by turning off all the lights. So your description of how you carry out the experiment, I can't really go through exactly what you're going to say, but it should be basically what I'd said before. Now, predicting the results of your experiment. Okay, so something like this for your method, by the way. So you can adjust the light intensity by moving the light further and closer away from the plant. Make sure you're saying we can measure the rate of photosynthesis by counting the number of bubbles the plant releases in 1 minute. And then you say something about moving the lamp further away and repeating again. Right, so this would be our results of our experiment roughly. Okay, so I've changed the numbers slightly, the heading slightly, to look at, rather than looking at the distance of the lamp and the number of bubbles, we've taken what we actually wanted to find, which is the light intensity and the rate of photosynthesis. So if you can see, what is happening is that as the light intensity goes up, or as our lamp gets closer, the rate of photosynthesis goes up, which is what we'd expect. Okay, and if you think about what shape this would give on a graph, it would look something a little bit like this. Okay, so I want you to pause the video again in a second and answer these two questions. I want you to just discuss them with each other. So what pattern is shown by the graph and the data? So you don't write anything down for this yet. And what would happen if we started increasing the light intensity even more? So if it kept going like 60, 70, 80, 90, 100, what would happen to the light intensity, do you think? Okay, so just 30-second discussion, so pause the video again now. Okay, when we're ready to move on, we would see something like this if we continued. Okay, so the pattern is showing that it's as it goes up, as the light intensity goes up, the rate of photosynthesis goes up. So that's what we can quite clearly see, okay, on the early part of the graph. So if I just kind of highlight this bit here, okay, at the start, as the light intensity goes up, the rate of photosynthesis goes up, easy. But what's happening at this end bit here, okay, it's starting to level off, and that is to do with this. So if we look at this bottom bit, okay, what has happened as we've got to the end is that we've started to get to a point where it no longer goes up, okay, and we end up with a graph like this. So we end up with a graph that goes up at first, okay, so we have the same thing happening at the start. So as the light intensity goes up, the rate of photosynthesis goes up, and we get a straight line, a roughly straight line going up. But then eventually it curves off at the end. Okay, like this. It curves off and goes flat because something else is stopping the photosynthesis from happening. Okay, so if we're looking at why it becomes horizontal, something else must be stopping the rate of photosynthesis increasing. Now, think about what else could affect that. There's two other things that we've said: the levels of carbon dioxide or the temperature. So one of those two things is now what we call the limiting factor. So that's a really important sort of term. Okay, so the temperature or carbon dioxide, they are limiting factors. Okay, so they are the things that are now stopping the rate of photosynthesis going up anymore. So I want you to, so when you're answering these sort of questions, you're going to get asked, why does it level off? So it's because something else, i.e. temperature or carbon dioxide, is the limiting factor, the thing that's limiting photosynthesis from happening any faster. Right, so I want to pause the video again and answer these questions in your books. Okay, so can you sketch this graph? Can you explain what happens at the start? So when it's low, when it's down here, so the start means, if I just highlight that quickly, means this bit here. Okay, so that's the start. In fact, I'll highlight both of those to remind you of that. And what happens after a bit, after it eventually, what's happening over here. Okay, so after that, can you predict what a graph showing carbon dioxide levels would look like as well? Okay, so pause the video now and have a go at those questions in your books. Okay, so sketch the graph, should have done that. Explain what's happening at the start. We should have just said something along the lines of, as light intensity increases, okay, the rate of photosynthesis increases. So, quite simple. As light intensity goes up, the rate of photosynthesis is also going up. Right, what happens after a while is, okay, that something else either carbon dioxide or temperature is limiting photosynthesis. So something else, some other factor, one of the other things that affects photosynthesis is now the thing that's stopping it from happening any faster. Right, so onto the extension. Predict what a graph would look like for the levels of carbon dioxide. So for the levels of carbon dioxide, okay, done that one. It would look like this, okay? It's very similar to the light. So in this, if you look at the start again, okay, so if we're looking at the start, as our levels of carbon dioxide increase, the rate of photosynthesis increases. But at the end, okay, it levels off because something else is now the limiting factor. So this might mean that light is now the limiting factor instead of carbon dioxide, because we're still increasing carbon dioxide and again, nothing's going up. So if you increase both light and carbon dioxide, then it would obviously shoot up even more. But if we're just focusing on one, because as a good scientist we should be just focusing on one thing at once, it levels off eventually. Okay, so what I want you to do now for the last bit of this lesson, so I'm going to end this just after you've gone through these questions and I've gone through the answers. I want you to have a go at these questions. Okay, so can you sketch a line graph for carbon dioxide? And now it should be similar to what you look for light, apart from you should have different labels on the graphs. Then can you describe the relationship between carbon dioxide and rate of photosynthesis at the start, and then what happens eventually? and then why does it stop increasing after a while? So all that we've been talking about limiting factors. Okay, so we pause the video again, and then I'll put the answers up, and then that will be it. So when we're ready, I'll go through the answers. So I'll just put them straight up on the board, and then I'm going to leave you. Okay, so we should get these answers for this question. So you're going to pause it right at the end, and I'll just say that I'll hopefully see you back in school soon.